Welcome to Biology 72. This is Human Physiology. This is the textbook required for lecture. This is the 15th edition, and I strongly recommend that you use this current 15th edition. More information about course materials are on both the homepage, the syllabus, and the orientation. Please make sure you have all of your materials right away, meaning today, the day that you are starting your lecture, week 1 lecture module. You should already have your textbook. It could be an ebook, it could be a physical book. You should also have your lab materials. So today we are going to, right now, start with chapter 1. It is a smaller, this will be a smaller lecture than most of your future lectures, so that's good for your first day. Make sure that you stay on schedule. I know that I'm giving you links for all the lectures, and it's tempting to put it off, but it's a lot of material. Even today, it will seem very easy and straightforward, but students always end up missing points on their exam for this chapter because they ignored it because it seemed easy. So easy material can be deceiving. Make sure that you're staying up with it. There's a huge difference between understanding it on your own and being able to explain it to someone versus just following along with a lecture. Okay, so from day 1, I want you to really practice really good discipline and study skills. Okay, I want you to start off strong and do very well. So stay on schedule with the lectures. Also, a reminder in case you forgot or got confused, you must attend the lab session via Zoom or you will be dropped from the class. So even if you complete orientation, you still need to go to your lab section and check in. So chapter 1 is called the study of body function. We will be focusing on homeostasis and feedback control, but there is a lot of other material in this chapter that you should read. But today's lecture will be mostly homeostasis and feedback control. Okay. So what is physiology? Well, the word physis from Greek, it means nature, and logos, also the Greek word logos means study or knowledge. So physiology is the study of biological function. Okay, so just like biology is the study of life, physiology is the study of the function of life, right? So how does the body work to maintain life? So we have a huge emphasis on mechanisms. So we will learn a lot of processes or mechanisms in this class. And we will have times where we're studying at the molecular level. We're talking about cells, what's going on within a cell, how cells are talking to each other, how information is sent from one part of the body to another part of the body. So lots of molecular mechanisms. We will also talk about tissues, systems, right? Organs and organ systems. Okay, so how do all the tissues of an organ or different tissues within an organ work together, etc. Okay, so above all, when you take a step back, you want to know, how is the human body accomplishing this task? How are you able to lift your foot? How are you able to run away from danger, right? How are you able to digest your food? So homeostasis, I know you've learned this term before, and in general you can say that homeostasis is keeping the body in balance, and the fancy definition that you'll have to memorize, especially for lab, is that homeostasis is the maintenance of a state of dynamic constancy in which conditions are stabilized closely above and below a physiological set point. Okay, so I know that was a lot. You can go to your custom lab manual and you can see the definition for homeostasis that you need to know for lab. But for the lecture component, everything you need to know is in these slides. And then, of course, making sure you understand the slides, you will also look at your textbook, okay? All right, so intrinsic control. Homeostasis is maintained by two categories of regulation, intrinsic and extrinsic. So first we're going to look at intrinsic control. And this is your local built-in regulation, okay? So there's going to be molecules and other components within the organ that are receiving information and responding to regulate that organ, okay? So an example would be an overworked muscle, okay? An overworked muscle, it's been performing metabolism at a high rate, so it needs a lot of oxygen delivered to it, and it also needs the waste to be removed, the CO2 and other waste to be removed, okay? So once you have an overworked muscle, now it doesn't have enough oxygen and it has a buildup of waste. So this is our problem, that this muscle is no longer, it's no longer in balance. Homeostasis has been lost, and so intrinsic control to restore homeostasis to this overworked muscle would involve the signals in the nearby blood vessels are going to vasodilate those vessels. You're going to have blood flow to increase both oxygen being delivered and waste being removed, okay? And then homeostasis is achieved. Okay, so when you have your overworked muscle, there's going to be signals to the blood vessels that are within and right near the organ. And so, and the organ in this case is the muscle. And once those signals are received, those blood vessels are going to respond by dilating. So vasodilation of the vessels is going to increase your blood flow to that immediate or local area, okay? Now I know this is a little tricky because we could say the same thing on a larger level, but so the key here is to remember that when you're talking intrinsic, you're talking local, right, immediate area built into the organ. Okay, now let's talk about extrinsic control. Regulation comes from outside the organ. We usually will be talking about either the nervous system or the endocrine system when we talk about extrinsic control. And as you know, hormones are released into the blood and carried throughout the body. Only cells with receptors for that particular hormone will receive that signal to do something, right? To turn on or turn off some function. So hormones are able to circulate throughout the whole body, but only the target cells or target organs will be affected by that hormone. And this is a long distance communication, right? So it's extrinsic control. It's outside of the organ that you're trying to regulate, okay? Nervous system, also traveling a far distance, but instead of a chemical messenger, now you have an electrochemical nerve impulse, right? So nerve fibers innervate the organs that they regulate, and when they're stimulated, these fibers produce electrochemical nerve impulses that are conducted from the origin of the fiber, typically the central nervous system, so all the way, either all the way from the brain or from the spinal cord, and then it's going to travel all the way to its target organ. Okay, so this can be quite far distance as well. And these target organs, they can be muscles. That's what we tend to think of when we think of nerve impulses stimulating something, right? But it could also be glands to secrete something as well. Okay, and so we call those target organs, we call them effectors, and we're going to see that term again in a minute. And this is a really important way that we maintain homeostasis, right? So we're going to turn on or turn off some function by some organ, right, in order to regain or maintain homeostasis. That's the idea. Okay. So again, using an overworked muscle as our example, it's needing oxygen and it needs to get rid of waste. And so this extrinsic control would involve chemoreceptors in the carotid arteries receive this signal that oxygen's low, waste is high, and that's going to signal the brain and the nervous system to respond to this problem. They will do that by commanding the entire muscle to vasodilate. Instead of just the immediate nearby vessels, now you have the entire muscles going to get more blood. Okay, and then that will help to restore homeostasis. Okay, so both intrinsic and extrinsic control will happen at the same time, side by side, right? You can have one or the other, but typically you're going to have a combination of both so that you have very fast and fine control. All right, so hopefully you are still following along. Let's continue our discussion of homeostasis. There are 4 components. These components are a stimulus, a sensor, an integrating center, and an effector. Okay, so now I'll go back and talk about these, but I wanted to give you an overview. Okay, so stimulus. This is some change in the normal balance is going to be the stimulus, right? So this could be like body temperature, for example, that you're getting too hot or you're getting too cold. And so the stimulus would be the change in temperature, right? But any stimulus is any change in balance of homeostasis, okay? Then you have your sensor, and it's going to detect this change. It notices the stimulus, and it sends this information about the stimulus or about the change through what we call the afferent pathway. You recall that from anatomy. And it's going to send that to the integrating center. The integrating center then receives the information from the sensor and makes a decision. And so we call it establishes a set point. And what that means, and we'll talk more about set point in a moment, but the integrating center has to make a decision. Is the information that was sent from the sensor, is this cause for concern? Does action need to be taken? And if so, what action? Okay, and so it compares the information from the sensor to what it's supposed to be, what the set point is. And if you have a difference, you're no longer within your set point, then you will send information out through the efferent pathway to tell the effector to do something about it, and specifically what to do about it. So the effector is the organ that carries out the directions given by the integrating center, and the purpose of this is to restore homeostasis. Okay, so we will see some specific examples because I know this is all general and a little vague. I'm trying to give you the overview of each of these, okay? So I told you I'd talk about set point. Here it is. So you know body temperature is set, and in fact I believe I wrote this for you. Most people's set point for body temperature is 37 degrees Celsius or 98.6 Fahrenheit. These numbers are not precise for every person, right? Everyone can have a slightly higher, slightly lower set point, But this is on average. When you take a large amount of people and you find out what their set points are and you average them, this is what you get. All right, so at any given moment, a person with a set point of 37C could be slightly above or slightly below the set point. So this line here represents the set point, and this above and this below represents the normal range. So it's okay to be, 37.1, 37.2, right? So 0.1 or 0.2 above the set point. But if you go too far, then the body's going to receive information. I mean, it's receiving the information constantly, but once it receives information that it's gone above, a couple decimal points above the set point, then it's going to respond, oh, okay, now perhaps we're getting too hot, or if you were below, getting too cold. So we'll talk about that. But this is the allowed or normal range that the body temperature is allowed to go above or below the set point. And this concept is the same for all of these different internal conditions of the body. So you have things like your blood pH, right? Your blood sugar, right? We have all sorts of different conditions that we have to maintain within a range, a normal range, and that range deviates slightly above and slightly below a set point. Okay, so continuing with our example, body temperature, right? So throughout the day, your temperature could be slightly above or below the set point and still considered normal, right? It's within the normal range. So no response from the body is needed. This is normal, and you will not, this is not considered a deviation of homeostasis. You are within the normal range, so no action is needed by the body. However, what if you do go beyond this normal range? What if you get too hot? What happens? So hopefully you were answering me. If the body gets too hot, the very first thing you need to do is get vasodilation, all of the blood flowing as close to the surface of your skin as possible. At the same time, your sweat glands are stimulated, so you start sweating. And so the purpose of this is that as sweat cools off the surface of your skin, your blood will be cooled off as it passes by the surface of your skin. Okay, same question, opposite. What if your temperature drops below the normal range? What does your body do? So your body senses that it's too cold. So you're going to vasoconstrict. You're going to pull that blood away from the surface of your skin, keep it as internal as possible, keep it warm. Your muscles are going to start to shiver. This increases metabolism, and that metabolism from the work that your muscles are doing to contract and relax quickly, right? Shiver. That's going to produce more heat. So your muscles help you to produce more heat while your blood vessels pull away from the surface of your skin so that they don't get cooled off, right? And so that's a classic example of how the body will use a set point to determine if you're too hot or too cold, or if you've deviated too far from the set point, right? You will use a negative feedback loop to return to homeostasis. So negative feedback, right, is the major physiological response to a stimulus, or to stimuli, plural. Always the goal of negative feedback is to oppose the stimulus, because you don't want to deviate far from the set point. You want to maintain homeostasis. So examples that I already gave is the body temperature, right? Blood sugar, we don't want to go above 100 milligrams per deciliter. Usually 70 to 100 is the normal range. Okay, so when you're really hungry, and this is the part of why you will feel hungry, is when your blood sugar starts to get near or below that 70, right? There's other things involved with hunger. It's actually pretty complicated. We'll be talking about that when we get to our digestive system. Okay, heart rate. Normal resting heart rate, of course, is different for each person, and it depends on how fit you are. But the average healthy heart rate should be 70 beats per minute, right? You have your blood pH. I mentioned that earlier as well. And you might as well memorize this number now because you will be using it a lot, okay? So 7.35 to 7.45 is the allowed range, the normal range of blood pH. And we have systems that we will be studying that control to make sure that you don't go above or below this. And then if you do, we have an emergency response to correct that. Okay, so negative feedback loops. You have your sensor. It's going to detect some change, right, from your set point. It's going to deliver that information, and it delivers information through afferent with an A, and it delivers that to the integrating center. The integrating center decides what to do with that information. If indeed you have strayed away from the set point too much, then it's going to tell the effector what to do to correct that. And that process will continue until you're back within the normal range, okay? And so here you have your normal range, and once you've deviated from your normal range, your sensor becomes activated, and then of course you have to go through your integrating center, but then the effector will be activated to restore within the normal range. And once you're back within the normal range, then your effector will stop, right? You'll stop the effector because you don't want to end up going too far below your set point. Okay, so figures 1.1 and 1.2 explain what happens when you go too far above your set point and what happens when you go too far below. And essentially it's the same diagram, but instead of showing this where you're going above the normal range and being brought back down, it'll show the inverse of going too far below and being brought back up. Okay, so definitely you want to always study the figures that are shown in lecture. You want to read the captions and the explanation in your textbook. Okay, here's another one. So negative feedback loop, giving a specific example using the way that the body controls blood pressure, or one of the mechanisms at least. Okay, so when you lie down for a few minutes and then you quickly stand up, right? So you lie down for at least a few minutes and you quickly stand up, you're going to have a sudden drop in blood pressure, right? Blood's going to suddenly be pulled down because of gravity, and you're going to have to correct that to get it circulating evenly throughout the body. So you have a drop in blood pressure, and that drop in blood pressure is the stimulus. The stimulus, this drop in blood pressure will be detected by the sensor. In this case, the sensor will be blood pressure receptors or baroreceptors, and that's going to then send information. And so these are sensory nerve fibers. This is the afferent pathway, and it's going to send this information to the central nervous system, which is the integrating center. Specifically for blood pressure, the part of the central nervous system that is being used is the medulla oblongata. Okay, and I believe you already know this from anatomy as well. Okay, so you should be able to tell me that the integrating center is specifically the medulla oblongata of the central nervous system. Okay, now The integrating center decides what to do, what needs to be done, and it's going to use the efferent pathway to tell the effector what to do. And this will be motor nerve fibers to tell the heart rate to increase. Okay, so this is a really nice example because you have a different sensor, integrating center, and effector. Sometimes you'll have an organ that can act as two or even all three of these functions. Okay, so of course, once the heart rate increases, you'll have a rise in blood pressure. So this message to increase the heart rate will continue as long as the blood pressure is below the set point. Once you get back to your set point, then this stops. Okay. All right. Next. So let's see if you're understanding what a stimulus, sensor, integrating center, and effector are. So please tell me the stimulus, sensor, integrating center, and effector for the following scenario. Make sure to be as specific as you can. And of course, once I give you the scenario, you want to pause the video and actually work through the question, okay? So last summer, 8-year-old Charlene was walking barefoot on the sidewalk. She stepped on a sharp piece of glass, and her immediate reaction was to lift up her foot. All right, pause the video. Okay, so the stimulus, yes, is the glass, right? The sensor would be the pain receptors, and pain receptors are called nociceptors, okay? So the nociceptors send sensory information of pain to the integrating center, and that's the afferent with an A, afferent pathway, okay? So what's the integrating center? Well, we don't know specifically, but we do know it's the central nervous system, right? And then the effector is what your response is, right? So it says she lifted up her foot. So how do you lift up your foot? If you pick up your foot right now and you hold it up long enough, you will feel the different muscles that are being used, right? So you know the abdominals engage and the quadriceps contract so that you can lift up your leg. Of course, there's a lot of muscle groups involved in lifting up your leg, right? Your glutes and your hips, right? Everything works together to lift up your foot. But the main muscle groups, you could say abdominals and quadriceps. Okay. All right. So the idea here is that you are as specific as possible to tell me each of these stimulus, sensor, integrating center, and effector, okay? And you are reviewing your anatomy knowledge, right? Use it, use your anatomy knowledge. Okay, positive feedback. Positive feedback is going to amplify the change that's being detected, okay? So you're moving further away from the set point. So positive feedback is not typically used to maintain homeostasis, but we do have a few examples of positive feedback in the body. Blood clotting, right? So you know that blood clotting is a series of events that gets triggered, and you get more and more platelets, more and more clotting happening until you have a steady, solid clot and you won't bleed anymore. Okay. Oxytocin levels continue to rise and cause stronger and more frequent contractions during childbirth or parturition. And you also have a hormone called LH, and we'll talk about that when we get to our reproductive chapter. but we have a luteinizing hormone, LH, that levels will very, very quickly rise. And so we call it a surge because it's very quick. And that is what will cause ovulation to occur, right? So these are all examples of positive feedback. And so you get this increase, this amplification of change until you actually do achieve some goal, and then you do go back to homeostasis, right? So like the blood clot, once you make the blood clot and you stop bleeding, then of course healing processes will set in, but you will return to homeostasis because you're not losing blood anymore. Same thing with childbirth, you will return to homeostasis because you got the baby out. And same idea with ovulation. Okay, so positive feedback, not usually how we get homeostasis, right? So when you think of homeostasis or maintenance of homeostasis, you should think of negative feedback. Okay, so chapter 1 still has a lot of topics, and they are all review for you, right? Tissues, organs, and systems. And you should already know all of that information because it's review from anatomy. So I'm not going to lecture on it. I'm going to trust that you will take the time to review it. I've included slides to help you as you review. So read the slides, read the textbook, make sure you remember your anatomy. And you should do that for every single chapter that we go through. It will have figures and little anatomy reviews, and I don't necessarily go over them because it's expected you already know it. But of course, you're human and you probably don't remember everything. So make sure you take the time to remember it. You have to know your anatomy that you learned already. You will learn more anatomy when you go into your next program, presumably nursing. And then you'll be tested on that when you go to get your nursing license. So keep it up. All right, this is the end of our first lecture, and I look forward to getting to know each of you. Bye.